right, good afternoon. It's nice to have the sun out. I was prepared for a long rainy day and didn't need my umbrella, so good thing. Really the only announcement I have, I'm kind of hoping to get through most of chapter 15 today. That's our innate immunity. And then looking to next week into chapter 16, adaptive immunity. So, stay tuned next week, but probably our exam the week after will just cover through that chapter 16. Whatever we cover on the Monday, the day after Easter, that will not be included on Wednesday's exam. So again, so it'll be probably chapter 10, 14, 15, and 16 is what I'm thinking right now. If we get a little ahead of time, we might include some vaccine stuff on there, but at least through 15 and 16. All right, any questions from last time? Comments? Don't be afraid to jump in. We were talking about cells that are important in innate immunity. So we labeled all these cells last time. We finished last time thinking about the granulocytes. Those are our basophils, neutrophils, and eosinophils that have these granules in them. And we kind of finished with thinking about, we talked about the function of the neutrophils. Remember, that's 50 to 60% of the white blood cells in our bloodstream are neutrophils. So we have lots and lots of neutrophils. They're short-lived, but we keep replacing them. Many less eosinophils and basophils. And I kind of, rushed through their function. They have some overlapping function. And again, we don't have very many of them. We think at least eosinophils can act by phagocytosis, but they probably don't commonly do that. They're probably mostly acting by when they get signals to degranulate. And very often, if a person has an elevated eosinophil count in their blood, it most likely is indicative that they're fighting some kind of eukaryotic pathogen infection. It can be a sign of allergies as well, but if they're got some kind of parasitic worm, perhaps. And I didn't get to showing you this last time. This is a picture that I didn't post because I don't have the citation for this. I actually found this picture on Pinterest, which is kind of weird. And I couldn't find it anywhere else, but I thought this was really cool. It shows how the eosinophils are kind of following chemical signals to some parasitic fluke. And you would think, well, how can the degranulation of just, one eosinophil have any effect on some multicellular pathogen? Well, it isn't just one. Like you can see there's eosinophils covering the entire worm there. So when they're, all these digestive enzymes and things that are in those granules, inflammatory mediators, we'll talk more about what's in the granules acting on that worm. It's in trouble there. Okay. And basophils really kind of have the same kind of function as eosinophils. They're not considered phagocytic, but they're going to target the same kinds of things. All right. So those are our granulocytes. Our agranulocytes. Right, these, we'll talk first about the monocytes. They were also differentiated from that same kind of stem cell, the myeloid stem cell. Monocytes are agranulocytes that when they leave the bloodstream, and there's not, a lot of them, 3 to 8% of your total white blood cells in your bloodstream are monocytes. When they leave the bloodstream, and we'll talk about when they would do that, especially during inflammation, they're going to leave the bloodstream. They're going to differentiate into two different kinds of cells, macrophages and dendritic cells. So we're going to talk about macrophages a lot. They are, their main function is phagocytic. So we're going to look at the steps of phagocytosis in a little bit more detail, but basically, so if I draw, we'll call that a macrophage. That M, I don't know what Greek symbol that is. That's kind of a shorthand for a macrophage. Is it theta? data, maybe? Okay. So if there's a macrophage and it's ingesting some, say, bacterial cell, okay, so when it's going to digest, or sorry, extended pseudopods and endocytes host that bacteria, it's going to be in a membrane-bound vesicle. That vesicle fuses with lysosomes, which brings about, hopefully, the death of that bacterial cell. Okay, and then here I have that macrophages process and present antigen. Okay, so as kind of the last stage of that phagocytosis, there's receptors that get kind of added to that membrane, and that macrophage is going to present little bits of what it digested. All right, so there's some processing that takes place there. It's kind of attached to those receptors. These are called MHC class 2 receptors, and we're going to talk more about them later, but just as kind of an introduction, when we're talking about processing and presenting antigen, that macrophage actually presents some of its antigen on the outside, and then it travels through the lymph to come in contact with cells of our adaptive immunity, B cells and T cells. So that's kind of how our adaptive immunity gets kind of kicked into action. And we'll talk more about how that happens. Macrophages are also considered garbage collectors. So they are traveling throughout our body. We have lots of macrophages in lots of different parts of our bodies, the spleen, the alveoli of our lungs, lots of places. They're going around and just kind of cleaning up dead and dying cells. All right, so cells are continually being worn out and replaced, and our macrophages kind of clean that up. They are long-lived. So we talked about how neutrophils typically only survive a matter of hours, where macrophages are likely to survive months, more likely. So you might think, well, isn't that a disadvantage for a neutrophil to die right away? Not really. If you think about, neutrophils can be phagocytic, but they often act by, releasing these granules. And when those granules are released, yes, that's bad for pathogens, but it also causes some tissue damage to us as well. So we don't want a neutrophil living for months and continually degranulating in our body in kind of an uncontrolled way. We want it to do a little bit, it dies. If we need more neutrophils, then we can make more. Okay, so macrophages aren't acting in a way typically that's going to cause the host damage, okay, where sometimes neutrophils are. Okay, let's see. And lastly, they communicate with other cells by cytokine. That's supposed to be a Y. By cytokine signals. All right, so we are going to talk a little bit more, maybe at the end of lecture today or beginning of next one, about some examples of cytokines. But this is just kind of a general name for a signaling molecule. They're usually proteins secreted by certain cells, and other cells have receptors for those. So again, it's how cells communicate, basically. Dendritic cells, also derived from monocytes. These are also phagocytic. Here's a picture of a dendritic cell over here. It's kind of it kind of makes you think about a neuron, a dendrite, because, and that word, I think it means tree in Latin. So it reminded somebody of long branches or roots of a tree. But these cells are also really good at processing and presenting antigen. So they can present that antigen on those long, kind of spindle-like fibers. Okay, so processing and presenting antigen. Okay, so those are its two functions. And also mention that they are plentiful throughout the body. We mentioned that there's dendritic cells in our skin. So places like the skin, mucosal membrane, mucous membranes. Okay, so patrolling for invaders. Okay, and this YouTube video link that I posted at the bottom there, that's kind of a cute little animation that compares the function of macrophages and dendritic cells and talks more about this antigen processing. It's just a couple minutes long. They talk really fast, but it's good information. Okay, the other kind of agranulocytes, and these are, remember, derived from the lymphoid stem cells, are the lymphocytes, right? So these mostly function in adaptive immunity. We'll just talk about them briefly here in terms of an overview, more about them later. Apparently, when you stain these and look at them under a microscope, you can't really tell the difference between what kind of lymphocyte it is. Like you can't really tell if it's a B cell or a T cell. Lymphocytes make up 20 to 25% of the total white blood count, but they're mostly going to be actually in the lymph, not in the bloodstream. So they can kind of move back and forth. B cells, again, just kind of an overview here. They are going to differentiate into plasma cells. And it's plasma cells that make antibody. Okay, so much more about what antibodies are and what they do and how B cells get activated to do that later. But we consider B cells to be functioning, you might have heard the term humoral immunity. The body's humors are just fluids. Okay, so antibodies function mostly in our bloodstream. They can leave our bloodstream, but that's usually where they're going to start out. Okay. T cells, there are different kinds of T cells. There are cytotoxic T cells, there are helper T cells, and a couple other kinds. We're going to function mostly on the cytotoxic T cells and the helper T cells. They are going to function in cell-mediated immunity. And that just means they are, well, T helper cells have a lot of different functions, but the T cells that actually act, the cytotoxic T cells that actually kill other cells, they do that when another cell is infected with a virus, right? So that's just kind of a little overview of their function. There is one other kind of lymphocyte. Natural killer cells are considered lymphocytes. We're not going to mention them very much, but they are going to act mostly on virally infected cells. And they do so non-specifically. So we consider natural killer cells as part of our innate immunity. And again, we're not going to explore their function a whole lot, but there's one other point where we'll mention those briefly. All right, so that's the cells that we're mostly thinking about their function. Let's talk a little bit more specifically about this whole process of phagocytosis. We mentioned it a few times in a few different lectures, and you probably have, the gist of what's going on here. We're kind of looking at the stage of this in a little bit more detail. So our phagocytic cells, we mentioned neutrophils, macrophages, and dendritic cells. Okay, those are mostly our populations of cells that are function as phagocytic cells. Okay, so before they're going to endocytose some pathogen, first, they have to be in the place where the pathogen is. So how do they know where to go? They're chemotaxing towards that site of injury or that invader, they're following cytokine signals. Okay, so they have to get to the place where the action is. All right, so they're following signals to do that. And then they have to recognize that whatever that cell or whatever virus is worthy to be phagocytized. Right? It has to be recognized as something that's not self. And we think the main mechanism for that are these receptors on the outside of macrophages, neutrophils, and dendritic cells called toll-like receptors. And that name is not intuitive. It comes from a German word, toll, and they call these receptors toll-like receptors because toll is originally discovered in fruit flies. And when we discovered these in humans or mice, whichever, somebody said, hey, these are like toll. And so anyway, it's not an intuitive name. We often abbreviate it TLRs. Lots of abbreviations that you really just got to kind of recognize if you read a paper research in immunology. It's just full of acronyms that kind of it's a good idea to get used to kind of recognizing those. So again, these are transmembrane proteins of phagocytic cells. Again, here's our phagocytic cells there. So what do? They recognize broad-based patterns. In other words, they recognize things that invaders are likely to have, or molecules, I should say, that we are not. Okay, so we call these PAMPs. So that stands for Pathogen-Associated Molecular Patterns. All right, so what kind of things can function as a PAMP? We found about 10 of them in humans. There may be more, but we found about 10 so far. So some things like LPS, something in the outer membrane of a bacterial cell. We have a toll-like receptor that will recognize LPS. Okay, that's something that we would never find in our cells. So that's definitely foreign. Peptidoglycan, flagella proteins. Okay, so those are just some examples of PAMPs. So when the toll-like receptor recognizes one of these molecules, then it's kind of like, yes, it causes the phagocytes to now, attach and endocytose the pathogen. It also calls into effect lots of other aspects of our immune response. So it kind of like trips in a burglar alarm. Hey, I'm dealing with this. It sends out cytokine signals to bring in other aspects of our immunity. So inflammation, we'll talk about the benefits of that in a little while. Complement, those are proteins in the bloodstream that we're going to talk more about. Maybe even jump-starting adaptive immunity. So your book doesn't have a graphic that shows the toll-like receptors. I always like to have kind of a picture to talk through a little bit. So I got this from another source. But here's, so this is showing, say it's a macrophage, for example, or a neutrophil. Okay, so here's some toll-like receptors in the surface. It's, they're anchored in the membrane of the macrophage. And you don't need to, I don't have this memorized, like what toll-like receptor recognizes what, but this TLR4 that recognizes the LPS, the people that discovered that got the Nobel Prize in 2011. So this is fairly recently that we've kind of understood this, and, pretty important work. So, other things, various things that, again, might be present in pathogens that we don't have, various membrane cell wall components, that protein flagellin, we don't ever have flagella. Okay, so that helps the phagocyte recognize the invader. It will phagocytose the invader into some endosome. And then there are other toll-like receptors that get expressed inside of the cell on the endosome, like TLR3, TLR7. they are going to recognize parts of the inside of pathogens. So once the lysosomes are starting to chew up that invader, the DNA is released. So of course, the DNA from a bacteria or a virus is going to be very similar to ours, but there are differences. For example, the methylation. I always forget. So eukaryotes, the cytosines are methylated. Prokaryotes, it's typically the adenines. So that's just one little difference that the toll-like receptors might flag and cause, again, cytokines to be released to call other cells to help with the invader. Okay, so again, these are the PAMPs, the pathogen-associated molecular patterns. All right, broad-based patterns. So again, there's only like 10. We've discovered 12 in mice, only 10 in humans. This is not considered specific. When we talk about specificity, we're talking about a million different receptors that would recognize like a million different things. So kind of just patterns there. Okay, so here's your figure from your book talking about the events in phagocytosis. So it has a little bit of a transmission electron micrograph to kind of get you try to visualize what that might really look like, and then they just kind of sketched out the rest of the phagocyte. So these black little balls here, that's some kind of microbe that the phagocyte is going to endocytose. All right, so the steps here, it talks about chemotaxis and adherence. And of course, the toll-like receptors are going to help with both the adherence as well as the ingestion. Okay, and I wanted to make a little note over here. Anything that's going to help the phagocyte be more successful at ingesting that pathogen, we call that an opsonin. Okay, so it's kind of a strange sounding word. It might be a new one. Anything that's an opsonin is going to stimulate or enhance phagocytosis. Okay, so there are a couple things that we're going to talk about a little bit later. I just wanted to mention them right here that act as opsonins. There are complement proteins that can make the cell more likely to be successful in phagocytizing a pathogen. And also antibody can act as an opsonin. So what antibodies do, I'll try to draw one right here, right? They bind to the outside of the pathogen, and then the pseudopod of the phagocyte would have a receptor for the kind of stem portion of the antibody, so it can kind of grab a hold of the pathogen a little bit easier in order to ingest it. Okay, so more about those two things later. So here, now that the pathogen has been ingested and it's surrounded in some kind of a membrane, it's been endocytosed. Now here we've got the lysosome that's going to fuse with the phagosome, and then we call it a phagolysosome, right? So what kind of enzymes are bringing about the death of that pathogen? What kind of enzymes does the lysozyme bring? Let me just skip to the next. I'm not sure where the next slide. I'm not sure if I meant this number 4. I guess that goes with the previous one. But what kind of things are within that lysosome? I'll skip back to that other picture in just a minute. There's about 40 to 50 different enzymes that we typically find inside the lysosome. I should mention, I should have typed this in here, but usually within the lysosome, the pH is pretty low, okay, about pH 5. So it's actually actively pumping protons into the lysosome. It does that, it spends energy, ATP, to do that. So active transport, it's pumping in protons in order to lower that pH. So that alone makes it an inhospitable place for most microbes. And then here's just an example of some of the enzymes that we might find in the lysosome. So lots of digestive enzymes, ones that are going to break down proteins or lipids or nucleic acids. Lysozyme, we've talked about that enzyme before as one that chops up those sugar subunits of peptidoglycan. So it's going to act on bacterial cell walls, and lots of other ones. So I was thinking today, well, why is it? Let me just skip back just for a second here. So at the end of phagocytosis, basically the pathogen is killed and the bits and pieces are eliminated and, secreted outside of the phagocyte. And I thought all those nasty enzymes in the lysozyme are now being released outside of the cell. So you would think that might cause some problems. I was just reading today that most of these enzymes, the digestive enzymes, they only work, they're only functional at low pH. Oh, that's supposed to be a 5. Okay, outside of the cell, where the pH is going to be more neutral, more like 7, those enzymes don't function anymore. So that's pretty ingenious, actually. And we're only having the enzymes work where we want them to. There are other enzymes that do some pretty cool things, and just a couple examples. There are enzymes that actually produce toxic forms of oxygen. We talked about these: superoxide ion, hydrogen peroxide, hydroxyl radicals. These are all oxidizing agents. So they're trying to fill up their outer shell of electrons. They're grabbing electrons from other molecules and causing lots of damage. There are actually enzymes in neutrophils called myeloperoxidases that actually are able to produce hypochlorite. Hypochlorite, if you remember, is bleach. Okay, so from hydrogen peroxide and chlorine ions, they can actually produce a little bit of bleach. So how effective? 99% of the time, whatever pathogen that is going to be killed by the cocktail of enzymes that are within the lysosome. Now, some pathogens have evolved ways around that. We talked about virulence factors, but most of the time that's going to be pretty effective. And I just wanted to add that especially macrophages have 2 levels of activity. So they have kind of their normal level of activity where they're just patrolling around our body and, again, eating up dead cells. Called them garbage collectors. So I couldn't resist the Pac-Man analogy. All right, so a macrophage is the Pac-Man. He's going around eating up the dots, cleaning up dead stuff. Sometimes that's not quite good enough. Sometimes we need kind of an extra level of activity there. So sometimes we know that especially macrophages can become activated. And they do this by getting the right kinds of cytokine signals. And when they're activated, it's like Pac-Man's eating one of the big dots. All right, so when Pac-Man eats one of those big white dots, what do you know happens? He gets bigger, he moves faster, and now he can even eat the ghosts, where he couldn't do that before. All right, so now our macrophages, once they get activated, they get bigger, they're moving faster, they're making even more lysosomal proteins. They have to burn through a lot of energy and increase their metabolism in order to do that, and they're going to produce lots of cytokine signals to other cells. So it makes them more effective at causing the destruction of invaders. The downside is that there is a little bit more tissue destruction as well. So all these cytokines that it's making are going to act to increase inflammation. And get too much inflammation, that's bad. So there is some cost there. Okay, trying to remember. I actually don't remember what YouTube video that is, so I have to go back. Oh, Oh, that goes to the slide, right? Okay, so this is really a great little animation. I wish I could play it right now. If you're able to click on this link, when I clicked on this link last semester, I couldn't get it to work, but I did it yesterday and it worked just fine. But it's like the first 40 seconds or so of this video. These are macrophages that have been activated, and then they've been filmed in time-lapse photography. So you can see them moving around by amoeboid motion, and they're enveloping these little yeast cells. So it's kind of cool. Again, it's really short. And that's an activated macrophage. Okay, question? Good question. Did we already talk about cytokines are and where they come from? No, we're going to talk about them a little bit more kind of briefly. But yeah, stay tuned. All right, so before we get to a little bit more on inflammation and cytokines, I want to talk a little bit about another aspect of our innate immunity, and that are complement proteins. And this is probably people's least favorite because it does look rather confusing when we get into the details of this. Try to keep in mind the big picture, just kind of what these proteins are, and we'll get, again, a little bit more detailed as well. But the name complement, it's kind of a weird name for something, but these are proteins. It's a series of about 20 different proteins that are present in the blood. They're synthesized in the liver. Okay, so these are proteins that complement lots of other aspects of our immunity. So most of the time, the proteins themselves aren't going to bring about the death of any kind of invader, but they're going to help other aspects of our immunity. They're going to help phagocytic cells be more efficient in phagocytizing. They're going to help increase inflammation that helps get cells where they need to go. So I think that's where the name complement came from. So we consider these proteins part of our innate system because they're not specific. Like the second time the complement proteins are activated, it's kind of the same reaction as the first time. And these proteins, we consider it a cascade reaction. So that means that one protein causes a change in the next protein, that causes a change in the next one, and the next one, and the next one. So they kind of activate each other, you know, kind of like a boulder rolling down a hill, it hits another one, it causes another one. Okay, so three main functions of complement proteins. Okay, I mentioned this before. They can act as opsonins. Okay, and I'll show you more specifically how that happens, but they're going to bind to pathogens so that our phagocytes are more likely to be able to adhere to them and phagocytize them. Okay, they are going to increase inflammation. And I know I always think about inflammation as being a bad thing, but when it happens in a very controlled way, it's a good thing. It helps, again, cells get where they need to go. So increases inflammation, and also we'll say chemotaxis of cells. Okay, so immune cells moving to the sites of infection. And lastly, complement proteins can act to kill an invader directly. And that's by the formation of a membrane attack complex. And of course, we abbreviate that and call that a MAC, membrane attack complex. Okay, so we'll see exactly what parts come together for that to happen. Okay, so here's this figure's not in your book. It's just kind of a little overview. We're going to talk kind of lastly about how this cascade gets activated. There's three different ways that these proteins can be called into action. no matter which of the ways it gets activated, we have the same result. So it's not like, oh, if it's activated this way, this happens. And if it's activated this way, something different happens. But one way it can get activated is if an antibody recognizes some antigen. Okay, we haven't talked about what an antibody is really and what an antigen is. Antigens are things on some kind of invader. Okay? The antibody is what our body produces. So when there's an antibody complexed with an antigen, that first complement protein, which we call C1, recognizes that. And then C1 causes other proteins to split. Those bind together, cause another one to split, kind of so on and so forth. Some of these bits here have specific functions that we'll talk about. But they can all kind of end up, or some of those complement bits are going to kind of line up in the outside of a bacterial cell and form a pore. Okay, and then the turgor pressure basically can cause that cell to swell up and burst if you get enough of those. Kind of the same way that like a bacterial cell, if the cell wall gets weakened by penicillin, it can eventually burst, it being in a hypotonic medium and again, too much water flowing in, right? And well, I might as well mention it here. That's more likely to happen for gram-negative bacteria because gram-positives have such a thick layer of peptidoglycan that those proteins can't very often punch all the way through that peptidoglycan. Okay, so these are going to punch through the outer membrane, that thin peptidoglycan layer, and then the cell membrane of gram-negative bacteria. So the membrane attack complexes are really only for gram-negative bacteria. Okay, so here's the little more detailed graphic that your book gives. And I've tried to be really intentional in your learning objectives about what I want you to know from this, and other things maybe you don't need to worry about so much. So again, it looks complicated here. I'll make a note of the things that I think are especially important. But if we walk through this, here's complement protein C1. And this happens to be the classical activation. So when complement is activated classically, that involves an antibody bound to some antigen, say it's a bacterial cell. Okay, and that causes, that complex causes a couple other proteins to split. So the C2 complement protein and the C4 split. When any of these proteins split, then the fragments we label C2A and C2B. All right, when C4 splits, we label that C4A and C4B. Okay, so the C1, basically when it's activated, it acts as an enzyme and causes the splitting of these other proteins here. Some of those fragments bind together to form basically and act as an enzyme. The C4B and C2A. I'm not concerned that you know like which part of which one. It's one of the C4s and one of the C2s. But that molecules is important enough that we give it another name. We call it C3 convertase. Okay. Because it splits C3. All right, so we got a lot of splitting. When C3 splits, it splits it into C3A and C3B. Okay, those two bits are important. They have a specific function. Okay, so I'm going to write that again up here. C3 acts, C3A acts to trigger inflammation. Okay, so we're going to see shortly how it does that, but it triggers an inflammatory response, and the inflammation that goes kind of hand in hand with chemotaxis. So it allows other cells to come to the site where there's some kind of attack going on. And then the C3b acts as an opsonin. Okay, the only thing I don't like about this picture is that it doesn't show that very well. So actually, C3b comes back up here and binds to the pathogen. So you could think here B equals binds. Okay, fragments that have that B, they're going to bind to something else. So it binds up here to the pathogen, and that again helps the pseudopods have something to hang on to, and is going to be more likely to phagocytize that pathogen. A good analogy is, if you've ever been on the climbing wall here at Grand Valley, I don't know, it's in this building somewhere, I forget. But you know, you can't climb the wall unless you got something to hold on to. So this is a foothold or a handhold if you're on that climbing wall. Okay, and you could think about A as, I always think A for allergy. Allergic reactions have lots of inflammation. So that's kind of how I remember which of those is which. Some of that C3 though binds to kind of leftover bits from here, the C3 convertase, and acts as an enzyme then to split C5. Right, so we get C5a and C5b. C5a again triggers inflammation, that A, allergy, and C5b also binds back to the pathogen, but now it's going to bind. So here's C5b right here. It's going to team up with C6, C7, C8, and lots of C9s to make that membrane attack complex. Okay, to poke the hole in that gram-negative, outer membrane, peptidoglycan, and cell membrane. Okay, so what do you need to know from this? You should know the function of the C3 A and B, the C5 A and B. You should know which complement proteins form that membrane attack complex. Sometimes I like to ask a question like, well, what if a person can't make complement protein C5a? Well, how will that affect, the immune response? So, well, if it doesn't have C5a, it's not going to have as much inflammation, something like that. Okay. In terms of, you wouldn't necessarily need to write this whole thing out, but these intermediates are important. And of course, understanding that kind of the end game here, or the end result, is that membrane attack complex. And you might think, well, poking one little hole in a bacterial cell, is that going to really cause a problem? Well, here's a picture of some gram-negative bacteria. Again, remember, gram-negative. It's got a problem because it's got lots and lots of membrane attack complexes here. All right, so the pathogen's not going to be able to survive with that many holes poked in its outside. So that hopefully kind of clears up why that's going to be such a problem. All right, so the last thing to think about then is how this protein cascade gets activated. All right, and there's 3 ways to activate that. We already talked about one, and this was the first one that was described, so that's why we call it the classical pathway. That involves antibody. Or you could say activated by the antibody binding to the pathogen. Now, you could think, in terms of, well, which one? Well, we'll get to that in a minute. Never mind. Okay. The alternative pathway, this activates the complement cascade. Let's say it's activated by pathogens. And I'll put in parentheses here that C3 is naturally splitting at a low rate. Okay, so so to put that a different way, it turns out that C3 doesn't necessarily need to be acted on by C3 convertase. It's just naturally breaking in half all the time. And if it happens to be in the direct vicinity of some pathogen, as well as a few other factors that have to be present, then the whole rest of the cascade gets activated. Okay, and it has to be, you know, within like a minute, or the C3 A and B start to degrade. Okay, so we'll look at a picture of that in a second. And then there's this third pathway, the lectin pathway. We'll say that it's activated by microbial polysaccharides. Okay, so there are some sugars that are commonly on the surface of bacteria that we don't ever have on the surface of our cells. Okay. Okay, so this is another YouTube video. It's called a Help Hippo, I think it's called. It's another like drawn animation that just kind of sums up complement. And they really talk really fast, but if you watch it a couple times, it will make sense. So this is your book's figure that kind of sums up those different activation pathways. Okay, so like I said, the classical pathway requires some antibody bound to a cell. So when I talk about antibodies, we're kind of calling in some aspects of our adaptive immunity. So this is, you know, going to require you know, B cells to get activated to make antibodies. This is not going to be kind of the first response, like the first time you've seen a pathogen, you're not going to have antibodies there to that pathogen that's going to activate this. If it's maybe the second or third time, then, you know, maybe it could get activated that way. The alternative pathway, that's the one where I said that the C3 is just constantly splitting naturally. If it's in the neighborhood of some pathogen, there's certain receptors that it has to recognize on a pathogen, or certain kind of amino acids, I believe. If so, the C3b will bind, and if it's in the neighborhood of some other protein factors that we're not really going to worry about, okay, then that can activate the cascade. And that lectin pathway, I just kind of cleared that up here on this slide. Okay, mannose, that's the sugar that's found on pathogens that we don't ever have on the surface of our cells. And then lectins are made in our liver that bind to the mannose. All right, so when we have lectin bound to mannose, that can split C3 and start the complement cascade and get, you know, the C3a, C3b, and the C5a and the C5b and the membrane attack complexes. Okay. Questions about complements? All right. Again, I did try to kind of post, okay, here's especially make sure you understand these things about complement, and that's in your learning objectives. And it's really only just a couple pages in your book. So, you know, if you want to kind of hit that again, you know, read a few pages there, and that should be helpful. Okay. All right. So finally, we'll get to inflammation. And hopefully, again, it's hard to kind of talk about every aspect of immunity all at once. There is a really good video that's part of your homework that explains inflammation. It's one of Dr. Bauman's video tutors. So Dr. Bauman is the main author of your textbook. It's really good. It's like 4.5 minutes long. Don't watch that at like quadruple speed or whatever to try to get through your homework quick. It's really good. It's just a succinct explanation of why inflammation is good. So I highly recommend that. Again, you'll see that as part of your homework. Again, it's common to think about inflammation as being bad because we associate that with pain. But on a small scale, hopefully you'll understand that it's going to help mobilize immune cells. So help them, chemotax towards where they need to be, either to the site of injury or infection. Okay. And if you have some sort of wound, whatever you call it, fall and skin your knee, there has to be some kind of inflammatory reaction in order for healing to take place. In order for our tissues to regenerate, there has to be some inflammation. Okay. So symptoms, I should probably say signs and symptoms to use those words correctly, of inflammation. You're familiar with these. Redness. The Romans described that as rubor. You may be familiar with that term. Localized heat. And they called that calor. I think I'm spelling those right. Pain. It's another symptom of inflammation. And the Romans called that dolor, as well as edema. Okay, and edema is just a fancy way to say swelling. Okay, so we'll understand shortly how all those things come about. But here's a picture that illustrates all those things. It looks painful, right? This is a condition called chilblains. It's just a kind of inflammatory reaction of capillaries in somebody's toes to cold. So repeated exposures to cold. So even when they're not cold right then, like you can wake up in the morning and have your toes they're swelled up, they're red, they feel hot to the touch, and it's kind of painful, kind of itchy, almost kind of painful. But that's a good example of inflammation. Never had an allergy shot, you're experiencing all those things as well. Your arm heats up, swells up a little bit, you get the idea. In short term, it's a good thing. We call that acute inflammation. Chronically, it seems like our body does this more than what we need. That seems to be the prevailing notion. So we don't want to have chronic inflammation, and we're going to take steps to try to relieve that inflammation when it lasts a long time. What triggers inflammation? Well, here's one way that it's triggered. We've got these two complement proteins, these fragments, C3A and C5A, binding to some granulocyte. Okay, so a neutrophil or a basophil, or the third one, eosinophil. Okay, the binding of those complement proteins causes them to release their granules. And what's in those granules, chemicals that we refer to as inflammatory mediators. What kind of things are inflammatory mediators? I have some further examples written on a slide in a few minutes here. One example that probably everybody's familiar with is histamine. Okay, you get a cold, you might take a drug that's an antihistamine, right, to keep you from your nose swelling up and your nose getting runny. Okay, so histamine is maybe the more recognizable of those inflammatory mediators. Okay, what do those inflammatory mediators do? They are going to act on the blood vessels. Okay, so again, in response to some kind of pathogen, or maybe you have some kind of injury, before that injury, here's what your blood vessels should normally look like. Your arteries, your capillary bed, and your veins. Okay, and then say there's some kind of injury. If you're breaking the skin, typically your blood vessels would constrict right away because you don't want to bleed out. But then soon after, as inflammation starts taking effect, here's some inflammatory mediators, these blue dots. They're going to act on this capillary bed and dilate the capillaries. Okay, so we get what we call vasodilation. That just means that the blood vessels get larger and they get leaky. Okay, so that's going to increase the blood flow. It's going to increase the permeability of those blood vessels. So the cells and all those cells that we talked about that are in your bloodstream, they're going to be able to migrate out of the bloodstream into the tissues where they need to be in order to act. And hopefully get rid of the infection. Okay, so here's another close-up of one of those blood vessels. Okay, so increased vascular permeability during an inflammatory response. Okay, so normally, here's a normal permeability of the venule, the one on the left. All right, so you've got red blood cells, you've got white blood cells, platelets, all bathed in blood plasma that are zooming along through your blood vessels. Normally, we do have kind of a small amount of, every once in a while you might have a monocyte, leaving the blood vessel and differentiating into a macrophage or dendritic cell. The red blood cells don't typically leave the blood vessel, but the white blood cells do occasionally. It's a complicated process of exactly how they do that, though. In the presence of some inflammatory mediator, you can see how this blood cell has gotten larger, and this interstitial space between the cells that line it, you can see it's gotten bigger. All right, so it's a lot easier for, say, this monocyte to leave the blood vessel and migrate to wherever it needs to go. This process by which, say, a white blood cell like this would actually leave the blood vessel, like I said, it's complicated, but it has to, number one, kind of slow down and adhere to the sides of the blood vessel. Okay, that's a process called margination. Okay, so a process by which white blood cells adhere to the side of the blood vessel. Okay, that has to be a series of different boxes checked in order for that to happen. So there has to be the right receptors recognizing the right, proteins. There's all sorts of things involved there. So that's actually complicated. Adheres to the side of the blood vessel, and then the process where the cell actually leaves the blood vessel, we call that diapedesis. Okay, so that's the process by which white blood cells migrate out of the blood vessel. Okay, so it's not going to be able to, it has to slow down and stick first before it can leave. It's not going to be going, really fast and just, like sharp turn and find the exact right place to leave. So margination and diapedesis. Okay. All right. So when fluid's leaving the bloodstream, it's going to accumulate a little bit and cause that edema. Okay, but I'm getting ahead of myself a little bit. If we look at those hallmarks of inflammation, hopefully now it's a little bit easier to understand why we get redness and why we get heat. That's due to increased blood flow. We get pain because those inflammatory mediators are going to stimulate the nerve endings. And when we get swelling, that accumulation of fluid can stimulate the nerve endings as well and cause a little bit of pain. Okay, that fluid that leaves the blood vessels and enters into the tissues, what good does that do? That's going to help dilute any kind of virulence factor that the microorganism produces. We talked about how some bacteria might secrete some extracellular enzymes or some toxins. All right, so it's going to dilute that effect. It's going to help clotting if it's some kind of injury. It's going to facilitate the quick movement of cells to the site of infection. So instead of macrophages having to move by amoeboid motion, it kind of go with the flow of that fluid. And sometimes, especially, that swelling can last a long time. So, you know, brought to my mind, if you have an athletic injury, you know, the dogma is always ice it, ice it, ice it. Or you have a hard workout and, you know, you ice afterwards. And that's supposed to quicken the time that your body recovers. But I've read some alternative views on that. That dogma, you know, there's some controversy there, because some people say that when you ice injuries, that just slows down the inflammation process and delays the healing. So I don't have the right answer there, but it's kind of an interesting controversy about, you know, the right way to treat injury, and, you know, when is inflammation good and when is it bad, and how do you make that distinction? I don't really know. Okay, so interesting to think about. So one other consequence of inflammation can be fever. And we say that something causes a fever, anything that causes a fever, we call it a pyrogen. So that prefix pyro means fever inducing. What kind of things can act as a pyrogen? And of course, I should say, pyrogens act on the hypothalamus. All right, so that's the region deep within our brain that controls, like, it's like the thermostat for our body. All right, so pyrogens are chemicals that act on the hypothalamus to kind of reset our temperature. What kind of things can be pyrogens? Oftentimes, there are cytokines that our immune cells would produce to cause our body temperature to increase a little bit. And that is typically done in a fairly controlled way. Sometimes a pathogen itself can secrete chemicals that act as pyrogens, and then it can be done in a very out of control kind of way. And then that can be bad. All right, so when the fever gets high enough, and I don't know what's too high, like 106, 105, something like that, you start worrying about the patient having brain damage. So obviously, when the fever gets up that high, you got to do stuff to bring it down, because that's not your body acting in your best interest. That's some pathogen, right? So if we think about, well, what good, if this is a, you know, side effect of inflammation, what good is it for our temperature to go up? Okay, well, here's some beneficial functions. We know that it can inhibit the growth or the reproduction of a pathogen. Now, most bacteria have a pretty generous range of temperatures that they can survive in. So if our body temperature goes up a couple degrees, that's really not going to have much effect on bacteria most of the time. So that benefit is probably slight most of the time. We do know that increased increased temperature can impede nutrient availability. And the best example I can think of there is in terms of iron. We talked about this a little bit their first line of defense, how there are, different proteins that can be secreted that will sequester iron to keep it unavailable for pathogens. So when our body temperature goes up a couple of degrees, that's a signal for lots of our cells to just kind of take up any available iron, and so it is not there for pathogens to utilize. So that can be a beneficial function of fever. We do know that it tends to stimulate our immune system. Okay, so when you have higher temperatures, you typically have faster reactions, again, up to a certain point. This can increase phagocyte activity, and make them work a little bit faster, and increase the mobility of white blood cells. Okay, and I would say that's a real thing. It's like we know that's probably the best benefit in terms of fighting a fever. And I would just add that it is kind of we'll say a barometer of health. And what I mean by that is if you have a patient that's, say, nonverbal or a baby, you take their temperature and it's elevated, oh, that gives you a hint that probably there's an infection taking place there. So that can be helpful to know. Occasionally, fever can be a side effect of cancer, but most often it tells you that there's some kind of pathogen at work there. Okay, so the question is, if somebody has a fever, should you treat it? All right, or yourself. If you have a fever, would you clear the infection quicker if you let the fever run its course? Or is it okay to treat it with some Tylenol or ibuprofen or something that's going to bring the fever down? And there's controversy about that, like most things. Most physicians say that it's okay to go ahead and treat it. That the discomfort that a fever causes somebody is, it's not enough to, speed up your healing, that you might as well just treat it because, all of these benefits really don't add up to a huge benefit. So you might as well make yourself a little bit more comfortable. And yeah, I admit to being that mom that withheld Tylenol, so my kids would just kind of lay down for a while. But again, so usually it's go ahead and treat it. It's not worth the discomfort. And remember, you should be fever-free without drugs for 24 hours until you go back to school or whatever. That's a rule that not everybody follows. But okay, so we are making good progress here. We're almost through our innate immunity. There's only really one other thing left here. I want to talk a little bit about cytokines. Okay, and really only one in particular. So sorry that this slide is so wordy. I didn't really want to take the chance to write a lot of this out or make you write it out. Mostly the goal of this slide, I just want to show you some examples of cytokines and talk a little bit generally about what they are. So you don't have to know these examples. You don't have to know what they do. Except for interferons, we're going to talk a little bit more in detail about. So cytokines, the term that we actually haven't used for all that long. The dictionary definition here, they're protein mediators that act as intracellular signals between cells. We used to call these interleukins. Okay, so when I took immunology, when I was your age, we just called all them interleukins, and there's about 30 or 40 different interleukins, IL-4, IL-5, IL-30. Okay, but we started to understand that these signals don't necessarily happen just between white blood cells, interleukins. Okay, they act on other different kinds of cells as well. So they decided, okay, this is a very familiar term, so we're not going to totally get rid of it, but we're going to say interleukins are an example of, signals that we're going to call cytokines. Okay, so just for example, I talk about cytokines a lot, and mostly, when it comes to our adaptive immunity, I'm going to say, don't worry which cytokine it is. But for example, IL-1, IL-2, a lot of those interleukins have overlapping functions. We know both of these activate macrophages. They can also help activate T cells and B cells. So just as an example of some of the specific function there. Another cytokine that you may recognize is called tumor necrosis factor. So TNF. Cool name. When they first discovered this, they named it that because when they gave mice huge amounts of it, with cancer, it's made their tumors die of necrosis. So they thought, oh, we've got the cure for cancer. But when they injected it into humans, it caused such rampant inflammation that, excuse me, people could go into shock. So I forgot to mention that before. When your blood vessels get too leaky and you're losing too much fluid, it can slow down your blood or decrease your blood pressure to the amount that you're not getting enough oxygen to your organs and everything starts to shut down. Okay, that's what we call shock. All right, so you get too much inflammation, the patient can go into shock and they can die from that. All right, so too much inflammation is bad. People that have certain autoimmune conditions like Crohn's disease or rheumatoid arthritis, that drug that's advertised on TV all the time, Humira, that's a tumor necrosis factor block. All right, so when you block that cytokine, autoimmune means your body's attacking itself. All right, so you try to block that, dampen the action of that particular cytokine. And then here where I list some other inflammatory mediators. So, there are cytokines that act on cells to release inflammatory mediators. So histamine is the one we mentioned, bradykinin, prostaglandins, leukotrienes, what exactly those molecules are. Some of them are proteins, some of them are not exactly proteins, but again, those are just examples of inflammatory mediators. You don't have to spit those back out to me. I just wanted to give you some examples there. And then I want to, in the last couple of minutes here, talk specifically about interferons. There's two different kinds of interferons. Interferon gamma, that works in adaptive immunity, and we're not going to worry about that function. But I do want to talk about interferon alpha and beta, because that's one of the only ways that our innate immune system has to fight cells that are infected with viruses. Okay, so your book has a little graphic here to explain a little bit how we think that works. Again, these are interferons alpha and beta. Whoops. Okay, and so here's a cell, some of one of the host cells that becomes infected with a virus. Okay, so you know what happens. Viruses, they take over the cells and machinery and make their own viral proteins. But before that cell gets totally shut down, it will start making interferon, and it secretes that interferon, and that acts on neighboring cells. Okay, this cell itself, there's nothing that can be done. It's beyond help. Over time, that cell is going to die, either by, lysis or whatever. There's no hope for that. But neighboring cells will have receptors for interferon, and that will cause the cell to make these inactive antiviral proteins. And if at some time later that cell becomes infected with a virus as well, then those antiviral proteins become activated and they bind to the cell's ribosomes in order to stop protein synthesis. And that seems like that would be bad for the cell, And it would be, but just for a few days, it can stop making all proteins. So then the virus is not going to make its own proteins. So it tends to, again, slow viral replication. All right, that's all the time we have today. That's good. We'll start with chapter 16 on Monday. Hope you have a good weekend.